Hi, Effie Nikki here. Today I'm going to talk about the male reproductive system. We're going to talk specifically about erection and ejaculation. So you should have already talked about the anatomy, male anatomy or the anatomy of the penis and all of the accessory glands and structures. Here, this last picture, I've shown you a sagittal section of external genitalia. Hopefully you've already talked about the scrotum, you've already talked about the epididymis and sperm production in the testes. But I just want to make sure that we know some of these key terms. So when we say copulatory organ, that's what the penis actually is. Its main function and goal is to deliver sperm to the female vagina, eventually sperm getting into the uterine tube, and hopefully fertilization will occur if that's your intended goal. So what's going to happen to the penis during erection? That's what we're going to talk about. We're going to talk about the two different types of erectile tissue that exist. And basically what's going to happen is this erectile tissue is going to fill with blood. The blood's going to become trapped. It won't be able to exit until after ejaculation. Not in all cases, but we're going to stick with the standard cases. And this engorgement of blood actually allows for the penis to become enlarged, to become rigid, and to actually do its job as a copulatory organ, which is deliver sperm to the vaginal female reproductive tract. Okay, so you've already covered most of this. I just want to make sure that we understand the process that the sperm have to go through before they even get out of the external urethral orifice. So how sperm travels through the male duct system, we know it starts in the epididymis. Let me get the pen going here. And hopefully it will behave. Okay. There we go. Close that. Okay, so we know that this is, let's try one more time. I hit the pen button. Pen, green, yay. Okay, so we know here we have, here's one, testis. We know that the sperm is going to be deposited in the epididymis for storage. It's also going to mature. It's going to travel through the ductus deferens to the ampulla of the ductus deferens. That's where the seminal fluid is going to meet up with the sperm, and they're going to both enter into the ejaculatory duct. Through the ejaculatory duct, it's going to travel through the prostatic urethra, then through the membranous urethra, and then finally through the spongy urethra and out, and that is the external urethral orifice. Orifice just means opening or hole. Okay, so we know that you've seen the structures before. You may not necessarily know what they do or how they function in the entire process. So epididymis, we just talked about a second ago. Let me point it out again. So the epididymis is this structure right here. Kind of looks like a pink croissant to me. I don't know why, but that's every time I see it, that's what I think of. This is where sperm are going to be stored and matured. So it takes several days for the spermatogenesis to occur, and you can imagine that the testis couldn't make enough sperm at the minute that you wanted it for it to be ejaculated out. So there has to be a buildup process or a storage process, and that is the epididymis. It's also going to have a maturation process. I'd have to look back at the videos. I don't know when they talked about spermatogenesis. Hopefully they talked about the maturation process, and that the sperm have to actually fold. That cytoplasmic inside of the cell has to actually wrap around. The mitochondria have to get organized so that they can be right there in the mid-tail section and propel the flagellum again. Mitochondria, lots of ATP production. Hopefully we've got that down by now. Okay, so during ejaculation, what's going to happen is the epididymis is going to contract. It's going to contract and push the sperm out into the ductus deferens. Somebody's probably already said this, but we're trying to stick with ductus deferens. If you've ever heard vas deferens, it's the same. We're just now getting away from eponyms, right? So things named after people or named in a more functional manner. So ductus, it is a duct that carries something to the outside, so ductus deferens. Okay. So what is the ductus deferens? This is the tube that's going to propel the sperm from the epididymis to the urethra. And it's going to go through several things before it gets there, and we'll see those in just a second. What's a vasectomy? A vasectomy is actual cutting or ligating of this ductus deferens. So you cut out a section. Well, where's the sperm going to go? The sperm can come up here, but then the sperm's going to go out this way. The sperm is never going to get this way. It's never going to be able to go through the urethra, to the external urethral orifice. So, and we'll talk about seminal fluid in just a second. Seminal fluid is made here in the seminal vesicle. For a man that's had a vasectomy, even though the ductus deferens is cut, his seminal vesicle is still producing semen. Therefore, there will still be something ejaculated once they have a vasectomy. There just won't be any sperm inside that fluid. More than you want to know, I'm sure. Okay, so let me go back to a different color. So we know that the sperm is traveling from the epididymis. That travels through the ductus deferens. It joins to this structure right here, the widening. This is the ampulla of the ductus deferens. And then the seminal vesicle that we just talked about, these two are going to join together, and that's called the ejaculatory duct. All right, so what about the urethra? I've already mentioned it's in 3 sections. Some teachers will make you know the 3 sections, others won't. You'll have to ask your individual instructor. But they make sense. They're pretty easy to remember because you can see that they're named after the sections that they would flow through. So if this right here is the prostate, that's called the prostatic urethra. So this part right here is prostatic urethra. This part is called membraneous or membranous, either way. And then this material right here is called spongy erectile tissue. And so that's why this section is called spongy, this section membranous, this section prostatic. So again, just a way to more precisely talk about a specific area. If someone was having a problem with the lining of their urethra, they wouldn't have to explain where. They could use one word. Prostatic urethra, we know it's the lining of the membrane that's in the prostate. And obviously something to note, men carry both urine and sperm through this urethra. It's kind of like a dub, but we have to say it anyway, right? And not at the same time. So sperm and urine are not going to pass through the urethra at the same time. And again, I've already mentioned urethral external orifice or external urethral orifice. I get it right. Okay. Come on. So once the sperm gets from the epididymis to the ductus deferens, to the ampulle of the ductus deferens, it meets up with the seminal vesicle, right? We just saw a picture of that. What is that seminal vesicle doing? That seminal vesicle is producing semen. So what is actually ejaculated? Semen. We'll see some other glands that come into play, but what's in the semen? Sperm. But is it just sperm? No. It's a bunch of other material. Fructose, so fruit sugar, same as glucose, just slightly different structure, but fructose, proteins, enzymes. It makes up 60 to 70% of the volume of semen or ejaculate. And you can see all these other chemical compositions here. We have an antibiotic. They're going to destroy bacteria. You have to remember the journey that the sperm's taking. So the sperm, first of all, has to get to the vagina, right? It has to come out the external orifice, get into the vagina. Well, the vagina is highly acidic. That's going to destroy sperm. There's going to be lots of bacteria because it is an opening to an external environment. So we're always exposed to bacteria. So there's going to be antibiotic properties. There's going to be prostaglandins that you see here. We're going to talk more about prostaglandins and some of the different roles that they play. But what's interesting here is, and again, I don't know if in the order how these go, if you've talked about cervical mucus or not, but there is a thick layer of cervical mucus, and the cervix is the part of the female between the vagina and the uterus. So the sperm have to get through that cervix to get out to the uterine tube to be able to fertilize the egg. So the prostaglandins that are in semen actually help break down that sticky mucus of the cervix so that the sperm can get through. Also, you see here reverse peristalsis. Maybe you talked about peristalsis before when we talked about like fluid flowing through a tube. Should have been mentioned in blood vessels back in 201 as well. But pretty much if you're trying to move fluid through a tube, it's this squeezing and it pushes the fluid out, and then squeezing and it pushes the fluid out, or food or mixture, or whatever you want to call it. In this case, the reverse peristalsis. So in semen, there's a component to help the vagina and the uterus contract more inward. So that's reverse peristalsis. So what are the sperm trying to do? The sperm are secreting a prostaglandin that's actually going to cause them to be carried farther internally into the female, where they can actually get to the oocyte. And there's also something called vesculase. It ends in ASE, so that should give you a hint that it's an enzyme. And it's something that helps sperm coagulate right after ejaculation. And again, staying in a clump, maybe it's better to be in that clump form to actually stay inside the vagina, because although the majority of semen does leak out of the vagina immediately after ejaculation. So there is this enzyme that helps some of them coagulate in hopes that they'll get to stick around a little bit longer in the female reproductive tract in case the egg is almost, ovulated or not. And we'll talk a little bit about that later, but it should have been talked about, or I don't know the schedule of your other lectures, but the sperm can live for about 2 days, and the oocyte is there for about a day to 2 days. So you have kind of this window of opportunity where the sperm might get there a little bit early, but if they just hang out and wait a little bit, the oocyte will be there. Okay, remember the next place that comes right after the ejaculatory duct is the prostate. So we saw the ductus deferens come into the ampule of the ductus deferens, the seminal vesicles also, and those two fuse together into, or empty together into, the ejaculatory duct. From the ejaculatory duct, it goes to the urethra that's inside of the prostate, or prostatic urethra. Well, the prostate also secretes several other enzymes, citrate or citrate, which is a salty substance. So you can read it here on the chart. I'm not going to read it off for you, but it secretes a more milky-like substance. It's slightly acidic. We have fibrolysine, which is going to liquefy the sperm. So again, once the sperm, they're going to coagulate upon ejaculation, but then they have to be liquefied so that they can actually swim to the egg. So they can't stay in a big clump. So there's fibrolysine. There's also something called relaxin. This helps in activating the sperm. And it's a little more technical than we want to get into, but pretty much the sperm have to be activated so that they start swimming vigorously towards the oocyte. And then the third bullet there where it says prostate-specific antigens. These are also similar to prostaglandins. So what we just looked at here, prostaglandins decreasing the cervical mucus for causing reverse peristalsis, same exact concept here with the prostate-specific antigen. The only thing I like to mention about this is because you will see these tests for men over a certain age, a PSA result, prostate-specific antigen. If this is what they use to test if you have an enlarged prostate. So if the prostate, it becomes really, really large, you're going to secrete lots and lots of PSA. So they can use the PSA as a judge of whether your prostate has become enlarged or not, if you're a male. All right, so only 2 to 5 mLs of semen are ejaculated, and that contains about 20 to 150 million sperm per ml. So let's just take the high number for the sake of it, 150 per ml, but let's do the low for the amount, so 2 mLs. So we're talking about 300 million sperm are ejaculated. It's quite a bit. The sad thing is they have to go through this torturous journey before they actually even get to the oocyte. So out of those 300 million, anywhere from 100 to a couple of thousand actually make it to the egg. Okay, the last accessory gland we're going to talk about is the bulbourethral gland. This is a pea-sized gland. You can see here it's at the inferior part of the prostate, so hopefully by now we know inferior is bottom, and that is the bulbourethral gland. Prior to ejaculation, it's going to secrete this thick, clear mucus, and the two principles for this. One is it's going to clean out the urethra because the penis and the urethra delivers both urine and semen. The urine has to be cleared before the semen can go through. And it's not necessarily like, oh, mixing's bad, but it's not great for the sperm. So again, the sperm have to take this very torturous journey, and what happens if there's any leftover urine? Urine is very acidic, so what could happen to the sperm? The sperm could be destroyed before they even get out of the external urethral orifice. Before they can even get to the vagina, they've got diminished numbers, if you will. The second thing is that it lubricates the glands of the penis. And sometimes there's people use, slang for this, and they call it pre-cum. I've heard that quite a bit in class. Oh, is that what you're talking about? Yes. So this is going to help lubricate the head of the penis. Hopefully you know by now the glans is the head of the penis. So maybe I shouldn't just assume that you know. Maybe I should draw it here. So this part right here, this is called the glans of the penis. And we also laugh about this when we talk about it, because the best analogy that I can come up with is that, have you ever been down a slide, or it's really, really hot outside, or if you try going down a water park slide with no water on it, can you imagine if the sperm are flowing through the urethra and they're getting caught every 2 seconds on part of the membrane or the lining of the urethra? Well, it's going to damage the sperm. They're not going to be able to move as fast. They may not get ejaculated. And the whole point of this is to fertilize the egg. Although sometimes that's not our end goal in nature, the way that we have to explain it for anatomy and physiology is that's the end goal, right? The sperm want to be able to fertilize the egg. So lubricates the urethra and the tip of the penis or the glans penis, and then neutralizes any trace of urine or uric acid that might be left over in the urethra. Okay, now to talk about the actual sexual response, now that we've gotten all the accessory glands out of the way. And basically, there's just a few things that you have to know about this. Erection. What is erection? Basic term definition for erection. It's the enlargement and stiffening of the penis from engorgement of erectile tissue with blood. There are 3 cylindrical bodies of erectile tissue. There's the corpuscle spongiosum, let me get it out, and then the corpora cavernosa. So the spongiosum goes around the urethra and forms the glands of the penis. So that's all spongy. The corpora cavernosa is here and here. And they call them paired dorsal erectile bodies. And let me close this. And you can see here, this would be the cavernosa. This is cavernosa. And then this part down here is the spongiosum. So oftentimes people look at that and say, oh, it kind of looks like a monkey face. So I always bring it up because it makes people laugh. All right, so these two are cavernosa. These are the 2 that are going to fill with blood. This one as well, and it has a different purpose, and we'll talk about it in just a second. All right, so what's going to happen specifically? Of course, we've got to get into the stimulus for the erectile tissue to fill with blood. It doesn't just fill with blood on its own. So you can see here sexual stimuli. What could that entail? That could be touch, that could be smells, that could be sounds, that could be mechanical stimulation. It can be induced or inhibited by higher brain activity or higher mental activity. So it's something that if you're thinking about it, it can happen even more. If you try to suppress it, you can do that as well. So it can be controlled. So the second bullet there talks about parasympathetic reflex. Hopefully we still remember something about parasympathetic and sympathetic. Sympathetic is the fight or flight. Parasympathetic is the rest and digest. And the parasympathetic reflex is going to promote the release of nitric oxide, or NO as they call it. And what is nitric oxide going to do? Well, so the parasympathetic message promotes the release of nitric oxide. The nitric oxide is going to cause the erectile tissues to fill with blood. And this is one of the few cases where you have a parasympathetic actually causing this kind of action. So it's a little bit different. Most of the time, like I said, parasympathetic rest and digest, we wouldn't really think of erection as rest and digest, right? So it is one of the unusual ways in which we use the parasympathetic nervous system. Okay, so expansion of the cavernosa. So what's going to actually happen is the blood is going to fill that erectile tissue, that corpora cavernosa. When the erectile tissue is filled with blood, it actually compresses or pinches off the drainage veins. So the blood stays and maintains that engorgement or stiffness of the penis. And when the spongiosum is filled with blood, that actually keeps the urethra open. So they have two different tissue types because they have two different functions, right? The cavernosa is going to fill with blood, compress the drainage veins. The spongiosum is going to fill, and that's going to cause the urethra to stay open. Again, the sperm have to be able to get out. So the urethra has to be able to stay open so that the sperm can be released through the external urethral orifice. All right, and the last word I have there is impotence. This just means you have the inability to maintain or attain an erection. So this usually has something to do with nitric oxide, and we can see there could be many steps here. It could be a parasympathetic reflex problem. It could be a nitric oxide problem. There could be something wrong with the erectile tissue. So there are a number of things, but usually what the problem is that it's the release of nitric oxide. So if a person is given more nitric oxide, they can usually attain an erection if they were having problems previous to that. But again, this is a lot of, you can look at the top part for sexual stimulus and that point that I made about this can be induced or inhibited by emotions or higher mental activities. You have to also keep that in mind. It may not be a physiological problem, it may be more of a mental problem. So however you want to think about that, it could be either or both. Okay, on to ejaculation. So basic definition, what is ejaculation? You're going to propel that semen from the male duct system. Another word that we use is orgasm. But I want you to understand the basic definition. What are you actually doing from a physiological standpoint, propelling semen from the male duct system? And this is caused by sympathetic spinal reflex. So this makes a little more sense, right? The sympathetic version of it, the fight or flight. This seems like an active thing, ejaculation, so that one makes a little bit more sense. And you can see all the things that are going to occur. The ducts and accessory glands are going to contract and empty their contents into the urethra. The bladder sphincter muscle is going to contract, so that stops the expulsion of urine. So, you know, one of those questions when they're doing sex ed, you know, can a guy, you know, actually urinate and ejaculate at the same time? And they probably didn't use those words, but you know what I mean. And the answer is no. The bladder sphincter muscle is going to contract, and that's going to stop the expulsion of urine. And then these bulbospongiosum muscles I have listed here at the bottom, these are going to undergo a series of contractions. So muscles in a very concentrated area, all contracting at once. Oh, that's it. We're done. We've talked about erection and ejaculation. Again, I would just go back and make sure that you understand the difference between the two, that you understand basic definitions, that you understand especially the parasympathetic versus sympathetic. One of the questions I usually ask is explain to me, explain the entire process, including the autonomic nervous system involvement in erection and ejaculation. So I'm going to move on and talk about birth and development, so I'll see you in a couple lectures. Bye.